Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello students. In the previous classes, we have started the discussion about biotechnology and its applications. In particular, we started with application of biotechnology in the field of medicine. The other applications of biotechnology in the field of medicine, they include production of insulin that we have discussed already. Now, one more important application of biotechnology in the field of medicine is gene therapy. Here, it is a very advanced application of biotechnology in the field of medicine. To define it, we need to understand first if a person is born with a hereditary disease. As the name suggests, hereditary diseases are those which are transferred from parents to the offsprings. One will think that these diseases cannot be treated, but with the advancement of science and technology, particularly biotechnological processes, it is possible to treat these hereditary diseases in an effective way, and one such approach is the gene therapy. First clinical gene therapy was done in the year 1990 to a 4-year-old girl who was suffering from a condition SCID, refers to severe combined immunodeficiency syndrome, that we shall take up later. Before that, we shall understand what is meant by gene therapy. Gene therapy, it is the collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo, which means if a child is diagnosed with some conditions or some gene defects, gene is responsible for production of some enzymes or hormones. If that defect is detected, then that gene defect can be corrected by gene therapy. How? In gene therapy, they will introduce they will replace a defective or non-functional gene by a normal functional gene. So this is a very simple way to understand gene therapy. Gene therapy is a replacement of a defective non-functional gene by a normal functional gene, which means here we have understood that if there is a gene defect identified in a child or an embryo, using gene therapy, that defective gene is replaced by the normal functional gene. That is using recombinant DNA technology. Recombinant DNA technology will help us to replace a defective non-functional gene by a normal functional gene. And that correction of the gene was done first in the year, clinically I am telling, not laboratory test. Clinical attempt was done in the year 1990 to a 4-year-old girl who are suffering from a condition called SCID, refers to severe combined immunodeficiency syndrome. Let us now understand how the treatment or gene therapy was done to 4-year-old girl using gene therapy method. To understand this, first we shall understand what is the symptoms and what causes SCID. SCID, as I said, it refers to severe combined immunodeficiency syndrome. Okay, so as the name suggests, so this condition is associated with immune system. So first let us understand how SCID is caused to a patient, or what causes the condition SCID. Here, SCID is caused due to deletion of a gene coding for an enzyme ADA, refers to adenosine deaminase. So in short, we can call it as ADA, which means if there is a deletion of gene coding for adenosine deaminase, then it will result in a disorder called severe combined immunodeficiency syndrome. Here, the gene coding for ADA is present on chromosome number 20. So, it can be called as autosomal recessive disorder. So, this you can expect in competitive exam. So remember this always. Gene coding for ADA, that is adenosine deaminase, is present on chromosome number 20. So chromosome number 20 is a autosome. So this disorder is also referred as autosomal recessive disorder. So now to understand how ADA deficiency causes SCID, let us understand the normal metabolism of adenosine in the normal individual. Just remember, I am explaining now what happens in normal individual. Then what happens later, I will explain what happens in the patient suffering from SCID. Now, in normal individuals, the DNA synthesis, or we can refer it as replication. For DNA synthesis, there is requirement of 4 nucleotides that we have know already. Nucleotides such as adenine, guanine, thymine and cytosine. So among them, one of them is adenosine in the form of dATP, deoxyadenosine triphosphate. Deoxyadenosine triphosphate in normal individuals, I am telling, in the presence of enzyme ADA refers to adenosine deaminase, will remove its amine group, one amine group, and converted into DAMP, that refers to deoxyadenosine monophosphate. Deoxyadenosine monophosphate in presence of enzyme purine nucleoside, here it is nucleoside, remember, nucleoside phosphorylase, it removes its phosphate group and deoxyribose. Deoxyribose means it's a sugar. and converted into a compound called hypoxanthine. Hypoxanthine, in presence of an enzyme xanthine oxidase, undergo oxidation, removed with the electrons, and converted into uric acid. As we know, uric acid is a waste. This uric acid is removed through the kidney. So now we have understood where does adenosine deaminase is required for. Where it is required for the metabolism of adenosine. That is, adenosine is present. It is not present freely. It is present in the form of deoxyadenosine triphosphate. For the metabolism of this, there is a requirement of adenosine deaminase. Which means if normal content is present, then it is utilized for synthesis of DNA. If excess amount of adenosine is present in the body, that should be metabolized. So that should be metabolized to removed through the excretory waste through the kidney. For its metabolism, there is requirement of adenosine deaminase. On the other hand, in normal individual, one more process is taking place. That is, there is presence of cells called lymphocytes. As we know, lymphocytes are the types of blood cells. Lymphocytes will divide quickly and undergo differentiation to form B cells, or we can call them as B lymphocytes, T cells, or we can refer them as T lymphocytes, and one more type of cells, NK cells. In expansion, natural killer cells. B lymphocytes will produce us antibodies, and these antibodies will bring about humoral immunity. And T cells and natural killer cells will bring about cell-mediated immunity. Which means the division and differentiation of lymphocytes is very much important for differentiation of lymphocytes to form B cells, T cells, and natural killer cells. So overall, it is required for the strong immune system. So now, it comes immune system. Here previously, what I told, ADA deficiency will causes the malfunctioning of the immune system. And malfunctioning of immune system will result in a condition, that is SCID, severe combined immunodeficiency syndrome. Now let us connect both. So what I told, in normal individuals, what happened? ADA required for metabolism of adenosine. Then this adenosine will be removed in the form of nitrogenous waste through the kidney. And in the same normal individual, lymphocytes will divide quickly and forms B cells, T cells, and natural killer cells, which build up a strong immune system. Now, what happens in SCID? The patients who are suffering from SID, that is severe combined immunodeficiency syndrome, in them there is no production of ADA. That is, adenosine deaminase is not produced. If adenosine deaminase is not produced, then dATP will not be metabolized. If there is excess of dATP is present, then it is not metabolized. That is, it is not converted into uric acid and excreted in the form of urine through the kidney. As a result, DATP get accumulated. It is not metabolized. As a result, it is getting accumulated. This accumulated dATP will inhibit DNA replication. So we have discussed in earlier concept, the same concept earlier what we discussed. There is requirement of adenosine, adenine, thymine, guanine, cytosine in the right proportion for DNA replication. If only adenosine content is more in the form of DATP is more and remaining are very less, then directly it will affect what? DNA replication. We know that for cell division there is the crucial step is DNA replication. If DNA replication is not taking place properly, then there is no cell division. If there is no cell division, then the different types of cells are not formed. Then you may ask question, why you are particularly talking about the lymphocytes, not generally about all the cells. Remember, ADA deficiency will affect all body cells. All body cells it will affect, but the more impact is on lymphocytes. So that's why we are focusing on it. So the more effect of ADA deficiency is on lymphocytes. Of course, it will affect all the body cells, but the more effect is seen on lymphocytes. See, lymphocytes for what they are required. Here we have discussed lymphocytes has to divide quickly to form B cells, T cells, natural killer cells. Then immune system will be strong. Here in SCID patients, there is no metabolism of ADA. It will get accumulated in the body. As a result, lymphocytes will not undergo differentiation. Then what happens? There is no strong immune system. So the condition is called severe combined immunodeficiency syndrome. The condition is very severe. It may lead to death also. Okay, since immune system is not functioning, then we are calling it the syndrome as severe combined immunodeficiency syndrome. Okay, so that is the case of SCID. And this SCID was first attempted to be cured on a 4-year-old girl in the year 1990. So to treat this SCID condition, there are various methods or there are various approach available. Among them, the first approach to treat the ADA deficiency is bone marrow transplantation. Bone marrow transplantation. Here, the SCID condition can be cured by transplanting donors' stem cells. If any person is identified with SCID condition, then from a normal individual, we can call them as donors. From donors, they will collect the stem cells and directly they will transfer it into patient. This is one of the approach to cure. Bone marrow transplantation is one of the approach to cure SCID condition. And second is enzyme replacement therapy. As the name suggests, here the enzyme ADA is injected into patient weekly. So here we know that ADA is an enzyme. Enzymes the normal enzyme, enzyme is isolated and it is injected into the patients. And whenever it is present in patient functional, for that time there is no problem for a patient. But after that, when once it is getting over in the body, then again they will suffer from the same condition. So periodic infusion of the enzyme, that is weekly or monthly, so however the doctor suggests, so that is periodic infusion of the enzyme is required. That is one more approach for treatment of SCID, that is enzyme replacement therapy. And the third approach is what we are discussing, that is gene therapy. And gene therapy is possible only because of recombinant DNA technology. So in the first lesson, we have discussed the procedure of recombinant DNA technology. Let us have a quick look at how gene therapy is conducted to cure SCID. So here what they will done is, first they will isolate lymphocytes from patient blood, and they will grow them, the lymphocytes, in a laboratory, in a medium they will grow. So let us consider these are lymphocytes. And these lymphocytes, to these lymphocytes, they will transfer a cDNA coding for enzyme ADA. So ADA gene is isolated, and that ADA gene's cDNA is introduced into the lymphocytes which are growing in a medium using a vector. That is, normally retrovirus is used as a vector. We studied in first lesson, retrovirus are the normal vectors used for transfer of genes into the animal cells. So I'll repeat. First, they'll isolate the lymphocytes from the patient blood and they will grow it on a medium. And to the grown lymphocytes, they will transfer the cDNA, which is coding for enzyme ADA gene. So in one more video, I will tell about what is cDNA. It's very important to know. Now this cDNA is introduced into lymphocytes using a vector retrovirus. So this may be asked for competitive exams. Normally you should know retroviruses are normal vectors used for transfer of genes into animal cells. So now these cells are called recombinant cells. Recombinant lymphocyte, let us say. So these recombinant lymphocytes are transferred or injected into the patient. So as we know, blood cells are not immortal. That is, lymphocytes are not immortal. Hence, patient has to have a periodic infusions. Periodic infusions of the recombinant lymphocytes is required in order to treat the patient. Now you may ask question, is there is no permanent cure for this condition? Yes, of course there is a permanent cure for the SCID if the condition is diagnosed in early embryonic stage, then in the early embryonic stage itself, the gene isolated from marrow cells producing ADA. So this is very important. Gene, marrow cells, they are nothing but stem cells. They have the capacity to produce ADA. That is, normal ADA gene is present in them. If those genes are introduced or those cells are introduced into the early embryonic stage, then this would be a permanent cure. So early approach, whatever we discussed, that is bone marrow transplantation or enzyme replacement therapy, even gene therapy, that is production of recombinant lymphocytes and introducing them into patient, they are not the permanent cure. Permanent cure is possible when the condition is diagnosed at early embryonic stage, and to that embryo, if they transfer the genes isolated from marrow cells directly, then it will become a permanent cure. I hope this video is helpful, and in next class we shall discuss about the molecular diagnosis methods. That is one more application of biotechnology in the field of medicine. Thank you.